Perfect. Sound is working. Great. I think we're live. Go for it.
Hey everyone.
We have share screen for the thing. Sorry, it's my touch. Give us 1 second, guys. Perfect.
Join the Zoom.
Oh yeah, I sent you the link on Slack. Excellent. He's in. Good. Other one. Go with the on screen. No, not me. Excellent.
Lovely.
All right.
All right. Now that we got that set up, I'm very excited to be here, guys. My name is Eric. I am a PhD student at Stanford in bioengineering, and I focus on novel architecture designs and their applications to vision, language, and biology. Go ahead. All right, we'll just jump into it. So I'm very excited today to talk about a new project and model that we call EVO. And this project is all about treating DNA as a language. And so if we think of a person as a book of DNA and genetic code, this book has 23 chapters of chromosomes, and it's made up of 3 billion characters of just 4 letters, A, C, T, and G. This simple vocabulary is responsible for pretty much everything you need to sustain life and to create you as you. It's appealing to think of language in this analogy, DNA as this analogy with language, because we all know that AI is pretty good at natural language, for example. And so things like ChatGPT are very powerful tools that understand, or presumably, a great deal about language, powered by things by the transformer, right? The kind of transformer architecture. And we've seen that as you train them and feed tremendous amounts of data and compute, they can learn a great deal about language. And so the great thing about DNA is that there's also a good amount of data, especially driven by the cost of DNA sequencing dropping dramatically from hundreds of thousands to hundreds of dollars now for being able to sequence in a person's entire genome. And so we care about learning from DNA because, well, it can help us understand disease better, perhaps better understand how to create new drugs, and discover new targets. And also this idea of personalized medicine. Just imagine if you can, for example, feed in an entire human genome as a prompt to ChatGPT. You could potentially query that genome for potential diseases, predict drug reactions, and just ultimately guide treatment options based on someone's specific genetic code. Now, we've got a long way to go before that can happen. But in this work, we focus on one particular area, which we're very excited about. And it's highlighted in this quote that I really like, which states that today we can, for all practical purposes, read, write, and edit any sequence of DNA, but we cannot compose it, right? So this is really getting at the difficulty in being able to design DNA. This was said by Francis Arnold, a Nobel Prize winner in chemistry. And so with that, context of not being able to design DNA, folks at Hazy Research, which is a machine learning group at Stanford, headed by Chris Ray, and Brian Hie, who's a new faculty member in chemical engineering at Stanford, we joined forces to come together and ask a research question, which is, can we design an entire genome from scratch using AI? Now, you might ask, why would you even want to do this? And perhaps naively, we thought if you could do this, then you could potentially create organisms that could target cancer cells, make better biofuels, or discover antibiotics. These are some of our pipe dreams that, to be clear, we didn't get to do, but we'll talk about what we did do along the way to try to reach this goal. So the plan for today is to talk about the challenge, why we think it's interesting to model DNA, and some of the innovation behind a new architecture we call Hyena to model and learn from DNA, and then share the results from what we found in the EVO project. So, the challenge for learning from DNA is that AI so far, in terms of their typical applications to biology, is that they didn't focus primarily at the single molecule level. And so we're getting pretty good at modeling proteins, even macromolecular complexes or RNA, in terms of predicting their function, their structure, but at this single molecular level. And so when you try to, for example, model whole cells, right? The cells are made up of these molecules, and then to whole organisms, right? Then it's orders of magnitude more complex than these single molecules. And so it's pretty daunting to say the least. But fortunately for us, in this case, in our approach, everything is encoded in DNA, right? So in terms of being able to build the molecules or your genetic code for an entire organism, it's basically encoded in our DNA. So the challenge is in biology, it's really encoded in somewhat different languages, as how we think of it. And so when we look at the central dogma of biology, which states that from DNA, it encodes information for RNA, which encodes information for proteins, which are the building blocks of life in yourself. Now, to be able to extract from each of these domains, it's been challenging. So what's commonly done is, or believed, is that you need specific models for each of these domains. So a separate one for DNA, a separate one for RNA, and a separate one for proteins. That's typically how we model biology or these domains. And so in this work, we are essentially saying we can focus on DNA itself because it's essentially encoded all of these information, right? It's just been hard to extract. That's sort of our hypothesis. So we compiled a large pre-training data set called OpenGenome, and to the best of our knowledge, it's the largest DNA pre-training data set with 300 billion tokens, and it covers prokaryotic and phage genomes, which are microbial lives, essentially. So lots of different bacteria, viruses, and this work was done by Matt Durant over at Arc Institute, who's an expert in these types of genomes, and we compiled them as their raw, unannotated form. So like we have no annotations, we're just training on raw tokens of DNA, and we train it like a large language model in this case. So the workflow is, right, we're going to sample from OpenGenome dataset a random DNA sequence, and then we're going to simply predict what's the next nucleotide, next token in the sequence, passing it through our EVO model, which I'll describe more. We can then take the pre-trained model, once it's done this essentially billions of times, right, through this training process, and apply it to downstream tasks that we actually care about. These are prediction tasks, but also generation tasks as well, right? We want to generate novel sequences that we'll describe more detail about. This is the overall flow. Now, so I mentioned what we want to model this foundation model, this genomic foundation model across the different modalities, right? DNA, RNA, and proteins. But we also want to model across scales. So this is another dimension that makes it challenging. So as I mentioned before, you typically model a single molecule at a time. But the cool thing is when you model across these different modalities, right, now you can start to model systems or groups of molecules from different modalities, because in actuality, in your body, it's more than just one molecule in one domain at a time. And then also to genome scale as well, to whole genome scale. That's the challenge we want to reach for. Now, we're not working in a vacuum. There are existing DNA foundation models that have done great work in this space and made a lot of headway. Things like DNA BERT or GenSLM or the nucleotide transformer. A key technical constraint that holds back, I suppose, many of these efforts is a couple areas that I like to highlight and dive deeper into, which is context length, which many folks are probably more familiar with these days, as you know, a lot of companies and models coming out for longer contexts. In DNA modeling, they typically model at just 512 to 4,000 tokens. for sense of scale, the human genome is 3 billion nucleotides, and so there's just a huge gap in terms of how much context length you potentially could want to use. This other dimension is on the tokenizer, right? So this is a key step in foundation models, where essentially we would dictate your resolution for your data that you want to model. And so in language, typically use BPEs or byte pair encodings, which will aggregate groups of characters into meaningful words, right? And so in DNA, it's similar, could be a similar process. You can also use k-mers, or like fixed DNA words. These are approaches that previous genomic foundation models have done, but we argue that there should be a different approach for DNA, and because of its differences from natural language, which I'll talk about. Focusing on context length first, we're interested in longer context in DNA, in part because single molecules, which you see here, can be relatively short, less than a kilobyte or kilobase, or less than 1,000 tokens essentially. And we have language models or on proteins that can fit these in context. When you want to look at whole genomes, right? So we have a lambda phage, which is the type of virus, or other types of gene clusters. Longer context matters to be able to model these types of molecules and sequences. And now I want to talk about what's the constraint on some of these models. Perhaps it's a familiar story that people have heard, which is a lot of these models, DNA models, use transformers like many other domains, and it can be restricted to context length because of the attention algorithm, right? The attention algorithm is this really powerful expressive operator, but it becomes at a cost, right? So as the sequence length grows, the time complexity, right, the amount of compute required will have this quadratic scaling or squaring law property associated to it. So if something doubles in length, then the computational need will quadruple. And so it can become intractable if it's quite a long sequence, particularly for DNA. I do want to contrast this with convolutions, which is another typical operator in deep learning, where it sort of has the opposite property, where the expressiveness, right, is somewhat smaller or lower because it has a local receptive field, so it aggregates information across a local neighborhood. But the trade-off is that it's much faster, right? So the time complexity is linear essentially, and so the question is there a way to get the best of both worlds? Something that's expressive but also very efficient. So coming back to the constraints of existing models, I do want to talk about the tokenizers, right? So the resolution of your data. And a key difference in language and DNA is that single character changes really matter in DNA. So you have things like SNPs, or single-nucleotide polymorphism, which show that in the right positions, a single character swap will mean a difference in what genes you have, what traits, what protein functions, and even what type of disease might be present in that cell. And so it's really important to have the high resolution in your tokenization. Another concept for resolution is this idea of frame shift mutations, right? So if you have a sequence of proteins, for example, that you're trying to model, if you just shift it by one character, the proteins that are encoded will completely change, right? And so having this aggregated tokenization sort of works against you in this way. And so the idea is, can we have a model that can model a single nucleotide for a single token? That's like the ideal case in DNA. The challenge is there is no large-scale language model trained at this resolution, none at the single nucleotide or byte level. And so this is a key constraint in terms of architectures, in particular transformers. And so last year we worked on a model called Hyena DNA, which is our first foray into applying new architectures or language models to DNA. Much smaller scale than the EVO project, but it's worth mentioning that the architecture here, because we did use much of the similar architecture. So what this paper focused on was two areas, right? The two challenges from previous models, which is a longer context. So we extended the context length up to 1 million tokens, which at the time was about 500 times longer than previous genomic foundation models. And then we modeled, or we tokenized at the single nucleotide level. So the two key contributions. This is a fun quote. I thought it was interesting to share. The Hyena DNA, got some buzz online. It was fun to see. And there's this AI minister from the UAE that caught wind of the work and said this quote, which said, from ChatGPT to Midjourney to Hyena DNA, it is now undeniable that AI is no longer a hypothetical technology, but one that warrants far more government time and resources across the globe. Thought that was flattering. Maybe it went to our heads a little bit. We got T-shirts to promote the work. But on the flip side, we also paid attention to folks who work in genomics, and we also saw comments that were perhaps more skeptical in healthy ways. But this one quote said, "For now, there is no serious evidence that DNA language models provide any gains in any useful downstream application. It's early days, so people should still keep working on it, but there's zero evidence that this is the future of genomic modeling." So we took that pretty seriously ourselves, and we kept on looking for grounded, useful applications. And that's sort of the backdrop to when we started the EVO project. And I'll just share a little bit of tidbits of what's going on as we were developing this. I do want to talk about the Hyena architecture itself and give a little bit of insights about its differences and perhaps some of its trade-offs for how we model DNA. So the starting point for the Hyena architecture is actually the transformer block, which is made up of the attention block layer and the MLP, as well as some residual connections. What we do for Hyena is keep the transformer block, but we swap out the attention layer for what we call a Hyena operator. And the Hyena operator's got two key primitives or operations that essentially try to mimic what attention's doing, but with faster operations. So the first is a long convolution, which tries to capture this global context that attention has, and the element-wise gate to capture what we call a data dependency operation, which I won't go into too much detail here, but we recommend diving into the paper for more details for that. I will go talk about how data flows through the Hyena layer. Let's say you have an input X. The first thing you do is project the input into three projections using a dense and conv layer. We call it the query key and value, just like attention, but we use them very differently. We take two of these projections and we do an element-wise gate. So this is literally just a multiplication per position. We take that output and pass it through a long convolution. And so what's a long convolution? It's like a regular convolution, except the kernel is very long or global. It's the same length as the input, which is in contrast to, I guess, the typical convolutions you think of, which are usually short or local range. We take that output and pass it through one more gate. And in large part, that is the Hyena operator. There are more details for the long convolution that I'll hint on, but again, point to the paper for more details. It is an implicitly defined kernel. So we use a separate function to parameterize the weights of the kernel. And then we do the convolution itself through the Fourier domain. So we do this through the FFT or fast Fourier transform, essentially to reduce time complexity from an O n squared operation to an n log n operation. This is backed by convolutionary theorem that I'll also just tease here for a second. And then more so talk about the model and architecture that we use for the EVO project itself. So before it was the first iteration, and then we using a somewhat upgraded version for this project based on the striped Hyena architecture, which is a hybrid model. So it's using Hyena layers plus a few layers of attention. It is a much larger model. It's 7 billion parameters, which is about 1,000 times bigger than the Hyena DNA model. And then we have this stage training to have more efficient context length extension. So we'll have first stage where we train at a short length of 8,000 tokens, and then a second stage where we extend it to 131,000 token context. So we chose this architecture after a good series of scaling laws experiments, which folks who are not familiar, this is typically done just in natural language to get a sense of how models train as you scale them in model size, as you scale them in compute and data, right? So the idea is that if you could model how these architectures train at a small scale, you can essentially extrapolate into how they might train when they're very large, because it's not always a given. So things can change as things scale. And so the idea is we're going to do this ahead of time to get a sense of what are the scaling dynamics and scaling laws, or what are the trends between different architectures that are prevailing in DNA modeling. And so these include transformers, but also newer architectures like Mamba, and of course the Hyena and striped Hyena versions as well. And so, what the main takeaways in the scaling analysis is that you get predictably improved performance in terms of pre-training and perplexity evaluation as you scale with larger compute, larger data sets, for example, which is not necessarily a given, right? Because we've only seen this happen in natural language, and so to see this in DNA, I think it's very interesting for us to observe. And the other takeaway is that there was a notable gap between transformers and also between Mamba and Hyena models, where the sort of the convention is that transformers are sort of the quote unquote the best architecture they use in multiple domains, but perhaps this could be different in DNA, for example. And so the paper is in review, and so some of the reviews that we got and comments in general is that perhaps it's too surprising or unbelievable that transformers are underperforming, and why is that the case? It's a good question. I think we're still trying to understand ourselves, but we do have some pieces that we can observe and share at least. So we have the set of experiments where we start with a transformer model, and then we gradually replace its layers with Hyena until you get to a full Hyena layer from left to right. And so what we see here is that as we do this swapping of layers, the perplexity is dropping all the way down to basically 90%, and that's one reason why we chose to keep a little bit of attention. But the trend is observed even as you just swap out one layer at a time. You can see this clear trend in improvement. And obviously, a natural question is still, why is that the case? We did have a follow-on work that focuses on scaling laws and new architectures, but on natural language. The connection there is that we look at synthetic tasks to really try to understand why these differences might be occurring in different models. And a couple key tasks or synthetic tasks that I thought were notable and perhaps had some similarity to DNA. And that's one is on noisy recall and compression. And the idea here is that in noisy recall, we're basically trying to see how well the model will perform recall or this task of looking up tokens in context, but we inject noise, right? And so in DNA, it's considered to be quite a noisy data distribution. And so the hypothesis is that being able to be really good at noisy recall or just filtering noise is particularly important in DNA versus language. Similarly, in the compression task, we're asking the model to essentially compress a sequence and then uncompress a sequence representation from a single token. And we think this is important for DNA modeling, particularly the single nucleotide level modeling that we do, because we're essentially asking the model to aggregate and learn how to compress meaningful DNA units through training instead of through tokenizing like language does. And so in this set of experiments we have, it sort of makes sense or provides a hypothesis into why like a convolutional model like Hyena might do better at DNA. Okay, so talked a lot about the architecture and what we're trying to model and some of the data. Now I'm going to describe the actual experiments and the training in sort of this timeline of emojis. The first one I call the pre-training and pray stage. And I call it this because there's, even with, some scaling laws to show there's an improvement on transformers with, newer architectures, there's still a lot of open questions like, is DNA too noisy of a distribution to actually generate, right? The perplexity that we see during pre-training is actually quite high, for example, and we tokenize at this byte level, right, as well, and have long context. So we're just kind of throwing all these constraints or requirements that are quite tricky, even in other domains or even large models that we've seen elsewhere. So yeah, we start training, we see the loss go down. We actually still don't know if it actually is learning like well, or if it's good enough to generate, right? We do compare with other architectures again at the 7 billion parameter scale. We see a lot of instability potentially, especially in transformers. We think because of the byte level tokenization or the single nucleotide tokenization, which has been observed in other papers as well. But pretty soon we get to the stage where I call the glimmer of hope, where we start probing the model to see what it's actually learning in terms of downstream tasks. The first set of tasks that we look at are protein function from a data set called Protein Gym, which is headed by the work headed by Brian Hie for this set of analyses. And the idea here is we're going to do a deep mutational scan, which essentially means we're going to take a sequence and we're going to mutate a random codon or random part of the sequence. And then we're going to look at the likelihood scores from the model, right? The output predictions of the model, and correlate that with some kind of fitness task. And in this data set, that fitness tasks look like protein stability, protein solubility, or binding affinity, which are important things for drug discovery, for example. In general, this method is done zero shot, so no fine tuning. It's sort of a clever way to probe the model and see what it's learning without having to fine tune. Main takeaway here is that against protein-specific models, right, models trained just on proteins sequences, the EVO model, which was trained on raw DNA, is quite competitive with these models. And so we're seeing that it looks like the objective that we set out to train on, which is a single nucleotide level, training from raw DNA, looks like we can grab a signal from protein sequences. Okay, there we go. All right, okay, so similarly, we apply this procedure to DNA and RNA as well. And in these cases, we can outperform existing models under this similar regime. In DNA, there'll be interest in zero-shot prediction of mRNA expression or protein expression or non-coding RNA expression for RNA. Still, you can still think of these as some kind of fitness value that you're trying to correlate your model's outputs and to see if it's sensitive at this single change or single token change in your sequence, which is quite interesting to itself as a task. Main takeaway again is the model seems to generalize across the three modalities or the modalities of the central dogma of biology, which to the best of our knowledge is a surprising and also first observation we've seen for a deep learning model as well. It's also useful to see where some of the failure cases, right? So the sequence shown before were all done on prokaryotes, which is what the model is trained on, so all in distribution. Now we start to see how it performs on human proteins, right? So these are proteins that are not seen during training for EVO, but in these other protein models, they have, right? So they have this advantage. And in those cases, yeah, EVO doesn't perform very well in those cases, but it's sort of understandable since it's out of distribution. That being said, we do have it seems reasonable that if we can improve performance on perplexity, right, during pre-training by showing it human proteins, that it should be able to learn human protein function as well. Okay, so now we'll talk about the next phase, where we started getting more excited about what we're seeing. And this is related to generation ability of the model, and also some of more of its longer range abilities as well. So EVO can design multi-molecular complexes. And this is beyond single molecules, which is what I was mentioning most models can do before. And I'll show some examples. I do want to show if this is going to work on the demo. So we put up an API for the model online, and someone was kind enough to make this cool wrapper, which essentially allowed you to generate a DNA sequence and then quickly pass it through AlphaFold as well, and so that you can in real time be able to see the structures of the coding regions, at least the DNA, and perhaps this can be quicker iteration time for design, which we thought was really cool. Okay, so more concretely in terms of what we were able to generate in this work, what you're seeing here is the first AI-designed CRISPR-Cas system. This is work led by Brian Hie on the project, and you might ask, what the heck is a CRISPR-Cas system? I've heard about it in the news. So I'll describe what it is, which is, you can think of it as a natural sequence or system in nature that bacteria have. It's a defense mechanism that they use against viruses. And so the idea is that bacteria will keep a copy of essentially invaders, like these viruses that will try to infect them. They'll keep a copy of this, invader or virus DNA in their own genome, so that when that virus comes back, they can recognize it and then they can cut it. They can cut it and essentially render it innocuous. And so what humans have done, right, they've sort of hijacked this molecular machine into useful gene editing tools, right? So these tools, these CRISPR-Cas systems, are made up of essentially a couple modalities, right? RNA and protein sequences. So this is currently out of reach of existing deep learning models because it has this multi-modality component to it that we try to overcome. So the idea is to try to design these systems that have RNA that will, you could think of it as the guiding or recognizing of DNA, and then the proteins which will actually do the cutting. So they work together to form this gene editing tool or system. And the idea is, we have CRISPR-Cas from Jennifer Doudna, who won the Nobel Prize for this. But turns out it's just one of a family of types of CRISPR-Cas systems with different functions, different modalities that they work on, different efficiencies and trade-offs in efficacy. And so we want to be able to discover new ones or create new ones synthetically, so that we can have more gene editing tools. And so this is a paper last year that specifically tried to do this. What they did was look out for existing sequences in nature and bacteria, and then literally just grab and copy those sequences and test them to see if they had cutting ability. For us, we're going to try to instead learn the distribution of CRISPR systems in nature, and then try to sample from it, and ideally sample novel ones that are not found in nature. That's the goal, we'll hope. And so we'll do things like prompt on a specific special class token, describing a type of CRISPR-Cas system, and then generate left to right the RNA and the coding sequences to create a full system. So part of the steps to see if you can design, right, you need to validate these valid sequences or these valid CRISPR sequences. And so we'll have a pipeline where we first check computationally, and then we're in the process of validating in the wet lab as well for actual fitness or function. So computationally, we can describe here in more detail. We'll look for things that real systems have, right? Things like tracer RNA, CRISPR RNA, or Cas proteins. These are just, you can think of them as like little widgets that need to be there to be a valid sequence. And so we'll use programs in computational biology, which could essentially try to mine or search for these in our synthetic ones to see if the presence of these modules are there or not. We can also check for folding scores from AlphaFold, or metrics like sequence identity, which is basically measuring how similar these sequences are in terms of the actual DNA characters and how many times they match, essentially. I'll skip that. For time, I'll jump to some of the other long-range tasks that we're pretty excited about. There's this one task called gene essentiality, and this is a test done normally in the wet lab only, which could take months to do. And the idea is to take a genome from a microbial life and then knock out a specific gene by adding a stop codon, a premature stop codon, which will essentially render that gene useless, and then see if the organism survives or not, right? So this is a way to see if that gene was essential for life or for this organism or not. And this is done in the lab. What we try to do instead is a virtual version of this. Can we do this in silico just by looking at the model's outputs? So we take a data set, which comes from experimental data sets, experimental results. We perturb a particular gene, and then we use the likelihoods again to see if it can be correlated or used as a threshold for essential or non-essential. And it turns out we can get pretty good performance above, at least above random, right? But above a baseline nucleotide language model. And then also, as we increase context length from gene only to 8K to 66K, we're seeing performance at least maintain in terms of being able to be sensitive to these single chain codon changes. And you get this essentially for free, right? We didn't have to fine tune the model to do this task, which is, again, a task that could take months in the lab, which is quite surprising to us. One of the last things we do is try to generate a whole genome sequence, right? So this is one of the earlier research questions that we had. And so here what you're seeing is a single sequence that we generate at 650,000 tokens. So quite long. And we start to probe to see, how realistic are these sequences? How realistic are the proteins that they make? And in this case, there are hundreds of synthetic proteins in this case. We group them by function in terms of similarity to what's seen in nature. I'll be a little quicker on this for time, but we can look at statistics in terms of how the coding density or how they're grouped in terms of where the proteins are in the sequence and compare them to those natural genomes. There's programs like CheckM that can assess for quality of sequences in nature found. We can kind of hijack that and use them to check the quality of synthetic genomes that we're generating as well. The main takeaway is that are these valid real genome sequences that we generated? Short answer is no. It appears like it's missing a number of different markers that would indicate a complete genome. And so what we like to think of this as what the model's learning is that it's learning a blurry image of what a whole genome is, right? So it's a step toward that global understanding, but some of the finer grained details like missing key marker genes, missing rRNA to actually be a real genome are missing. And so that kind of asks, the question, where does that leave us? Like, what are the next steps, right? And for us, we looked at some comments out in the Twitter sphere, right? Some folks have been thinking this is very promising, very exciting. Some people are confused, like, what's a foundation model even mean for biology? Does that mean you can design a dragon with a blue tail as a prompt and then generate a sequence to do that? I don't think so. But concretely, for us, the next steps are to improve the generation quality, right? There's a number of steps or tools in the language community that we can borrow from that are not even used at all in biology. And we think it's a huge a research opportunity for us, but also for just the broader community, right? There's things like alignment and feedback from reinforcement learning that can be used. Again, we're just using raw pre-training generation, right? And what we've seen in language, and especially things like ChatGPT, the alignment step is quite important for high quality sequences, right? This is not done in biology at all. So there's some room there. We're definitely interested in scale and also applying the model to more complex organisms. So we focused on microbial life because they're simpler life forms essentially. But mammals and humans perhaps are far more complicated. And so seeing how if they scale to similar performance as what we saw here is going to be an open question still. But whatever we do work on next, I do firmly believe that it's going to need to be at the intersection of two very distinct areas to be able to drive innovation, right? So there's folks on the AI side who believe that scale is all you need, right? Just throw more DNA and more compute at it, and you'll solve biology, right? And then there's folks on the biology side who are more agnostic to the methods. They're just like, architecture doesn't matter. If you just choose the right problem, then that's the real question. Like, what are the right problems? But I really do believe that it's going to be a mixture of both of these that's going to really move the needle and accelerate discovery. I'll move past this. And so going back to the final question about can we design an entire genome using AI from scratch? And the short answer again is no in our case. But I think what's really exciting is that what we show here is that when you go after those crazy big ideas, you can be surprised about what you find along the way that you can learn. I'll just close with saying that this was a very large team of folks in biology and medicine and machine learning, and so not possible without them. And then also quickly just provide some resources here for our code base, some Colab tutorials to try things out. The weights are public. We have a blog here that's a good entry point for finding these links, and happy to take any questions. Thanks so much.
Thank you, Eric.
All right.
I'll kick it off with a couple of student questions that we have in the pipe. One question from a student is, for the scaling laws, excuse me, besides the performance gains, are there any other emergent abilities or behaviors that you've seen, as you scale up these models compared to what the smaller models could do?
Ooh, yeah, that's a good question. So I'd say we haven't done enough on this part, I'd say. We have a lot of checkpoints along during training, but we've mostly evaluated the final checkpoints, except one where we did evaluate the zero-shot abilities of the model for protein fitness. And it looks like in some areas where the model did not as well on earlier checkpoints, in particular, if those samples are not well represented in the data set, if you train for longer, we saw their improvement jump quite a bit. So it looks like the model is, optimizing initially for well-represented samples, and then for less well-represented samples, it's improving later during training. That's one thing that we noticed.
Cool. Another question. What are the noises that, you talk about DNA is a very noisy environment. What are the noises that you worry about? Is it sort of, the differences in individual genomes, or is it more related to the SNPs, the frame shifts, like that kind of thing structurally?
Yeah, good question. So for the noise, people usually refer to the noise in DNA to be the areas of the non-coding regions. So in DNA, you have coding, which is parts that specifically encode information for proteins. And then you have the non-coding regions, which is sort of people kind of consider everything else. And so these can be like the regulatory regions, like they'll dictate how much and when to use the genes, basically. But those rules about how to regulate are way less deterministic. And so it's somewhat considered noise in that sense, but also there's also just uninformative regions where the noise could be just a bunch of repeat characters. So like you'll have like 1,000 long of just a bunch of the same characters, or regions that are just like structurally just there to like hold up the DNA, and they don't actually encode for not that we know of that encode for regulatory or coding regions. And so this percentage of noise versus coding regions sometimes considered is called as coding density. And so in humans, this ratio is like quite dramatic. Only less than 2% of your DNA has genes. The rest of it is regulatory non-coding stuff. And a good chunk of it in humans is noise, which is or thought to be noise. And so again, these repeat characters or just like parts of the genome that we have not yet found a reason for their function yet.
Another question. So you talked earlier about the zero-shot performance discrepancies across these different modalities of models. And so one question is, can these differences be explained by just the different amounts of data available for these different modalities, or is it something kind of more fundamental going on in terms of the architecture and design?
Yeah, that's a good question. I'd say that could be the case, but in general, I think there's a lot of potential factors that play a role. Because we don't control for model size or data, so we were not trying to necessarily say one model is state of the art over the other, because you have to control for a lot of things. I think what we're more so using these zero-shot tests to show or to see if the model can generalize across domains. Because what we're most interested in is a model that could a single model that can do proteins, that can do RNA, that can do DNA. And in our case, as long as it's competitive or, if it's great, if it's surpassing, it's just interesting to us or striking that it can even pick apart these things from each of these modalities because it had not been shown before, simply. And so that's the first question. And then, yeah, if you want to control and see if which model is better, yeah, you do want to control for same architecture size and same model. But then it gets complicated at that point because one set of models is shown protein coding sequences, another one is shown raw annotated DNA. So there's a lot of different confounding things. So there's more questions about that kind of comparison. So our purposes for using them are a little bit different than what machine learning folks usually try to benchmark on, I say.
Another one. Actually, this is a good one. What is the data that your alignment process would use in order to instill preferences into the model? But if you were to go down the alignment, that's a student question. That's not a new question. That's a question.
Yeah, that's such a good question. So we're actively thinking about what's a good way to incorporate some of these alignment techniques. But broadly speaking, so let's take a specific example in the CRISPR sequences. You have portions that are RNA, you have portions that are coding proteins like Cas. Some of the failure modes, right, that we observe is that parts of those regions, parts of their RNA, will be missing like key components, key modules essentially, or like the sequence will get truncated too early and just have a bunch of repeats thereafter. And so we can visually see those for one. We also have heuristic algorithms to check for the presence of these key modules. Things like prodigal that will tell us where the coding regions are. Things like checkm that will check for coding density. So we have these like these rough computational bio tools to check for how realistic they are. So in that sense, we can filter by these algorithms and then place them in a bucket of like, these are the preferred ones, right? And then on the flip side, these are the failure ones or the non-preferred ones. And then we can feed in something like, for example, DPO, which requires that sort of kind of paradigm, right? You need to have one that you prefer, then one you don't prefer, and then you train, for example. Michael, you want to?
I guess, I mean, yeah, so I guess like, I mean, in that case, like, right, like, so, maybe piggybacking on that question, that, so like, what kinds of, or how exactly do you consider sort of like, oh, like this is preferred, or this generation is preferred versus another generation is preferred? And like, are you seeing like promising results, I guess, with like the current setup?
Yeah, so in general, we're operating in a low positive regime or environment. So we don't really have the luxury of saying like we have two positive ones, but we just happen to prefer this one over the other. What we do now is we take all the positives right now, and then we essentially will mine for the negatives. That could be just like sampling a random one, maybe noising it to the positive ones to make that an unpreferred one. And so that's how we've been setting it up right currently. But yeah, we're very much open to like other people's ideas about what are good ways to find negatives or even more positives perhaps. But yeah, good question.
I mean, I guess like is all this generation, or when you're sort of generating sequences, you mentioned earlier, for example, that like there are certain, let's say, like structures that need to basically be conserved. Let's say in like, I don't know, Cas9 or something, right? Like there are certain things that are free to vary and they could be better in terms of like, I don't know, performance, you've changed something a little bit, but then there are other things that perhaps like should stay fixed, right? So like for generation, do you consider everything as just like, do you just generate everything at once, or do you consider sort of like keeping certain regions fixed and then only sort of like generating parts that are free to, I guess, vary based on the science we know?
Yeah, good question. So I think what you're alluding to, or how we think of what you're describing, is sort of the prompt engineering part of it. So like, which parts do you keep or fix and feed the model as a prompt essentially, and then what part of the model do you ask?
Or I guess mechanically here, this is just, I'm just curious, like when you do generation, right? Like, do you just generate the entire sequence, or do you ever play around with, let's say, only generating parts of the sequence and keeping others fixed? For example, like we know that, okay, like for some kind of like tertiary structure, like you need like this amino acid sequence. So like maybe it like doesn't make sense to try to like generate that part as well, because any variations could like be hallucinations basically, right? You don't want that.
Yeah, we haven't done that yet. So the short answer. So we will typically prompt on something and then ask the model to complete the rest. But maybe you're like wondering, will you just ignore parts of it, just like replace with a real sequence? Maybe you can do it that way. Yeah, it's not clear how we can do partial design, partial completion. At least in this like left to right kind of style generation for large language models. What you're describing perhaps more fits better like with a bidirectional kind of training style, like if you get BERT to generate, or perhaps it's your diffusion style, right, where you can like fix certain parts of the sequence and then like infill or outfill around it.
Yeah, like it sounds like maybe in this case like some kind of like mass language model, which would be I guess more aligned to this. And I guess that's also another follow-up then is like just, I guess, two follow-ups. So maybe one follow-up directly to that is like, did you consider sort of bidirectional things modeling, and like why the causal language modeling, I guess, like initially?
Yeah, it's a good question. So we consider it for sure, and it's definitely still on the table. I think it's a question of trade-off. So in general, it's not clear how you would do a mass language model generation version. So there's multiple ways. It could be considered kind of like hacky. It starts to look maybe even like diffusion a little bit. But in general, there's an efficiency trade-off when you're going to stray from like the causal versions. They're just most language models just optimized in terms of inference for this left to right style. And so you can kind of think of other versions of generation that could make more sense biologically or just from our first principles. But then there's going to be a trade-off in terms of being able to get these systems to work well, essentially. In short, there's basically a good question to ask is, why is every language model out there a causal left to right, right? So there's something trade-off going on in terms of keeping it that way versus other potential approaches. Not to say that those other approaches are bad. It's just that the simpler approaches, I suppose, or the more efficient approaches seem to be this causal left to right. So we thought it's worth exploring initially, but we're absolutely interested in more first principles driven generation as well.
I guess how exactly do you prompt? That was the second question.
Yeah, so for prompting, we in the results we described in the paper, we prompt on special class tokens. So that could be the species type if we're trying to generate a whole genome. And then for the CRISPR-Cas systems, there's basically different sub CRISPR types like Cas9, Cas12 or 13. They'll also have their own special tokens. And so we'll just prompt on those classes and then ask the model to generate.
So can I ask a quick question just as a non-bio person in the room?
Yeah.
Like, can you just talk a little bit about why being able to generate synthetic CRISPR-Cas9 systems is like an important metric? Because I get the sense that you have an intuitive understanding of why that's important that I do not have.
Yeah, that's fair. I also didn't really know why too. I thought you just needed one CRISPR-Cas system. But so folks are interested in discovering new versions of CRISPR-Cas systems because they have different trade-offs in terms of accuracy and efficiency as well. And there's new types of CRISPR systems that are focused on different modalities, right? So the CRISPR-Cas that you normally hear about from folks in Jennifer Doudna's lab, they cut DNA specifically, but there's also CRISPR-Cases that cut RNA as well. And so essentially people, drug designers or gene editing people or folks are interested in new CRISPR-Cas systems depending on their problem and use case. So it's like another tool in the toolbox that could be better at one problem versus another.
I see. And in terms of what would make it better, it would be like in very specific conditions, in this like kind of cell or something like this, it works better or something. I'm making this up, but I'm hoping I'm on the right track.
Yeah, this is perhaps beyond my expertise as well, but I do know that there's a recognition part to a CRISPR system, and so perhaps some systems are able to better recognize. Some systems are better at recognize certain parts of sequences, or cut certain parts of sequences to better accuracy.
I see.
So you can kind of think of it as like, at least with Cas9, it's kind of like a pair of scissors, right? And it's like you have like this like little like guide RNA, which is another sequence that says, okay, based on this sequence, you're gonna direct these scissors to like this part of your genome. But then even then, right, like the scissors are only as good as like what the protein folded has in some sense. So you could have like messy scissors, like, okay, although they're supposed to like bind to this part, they actually cause some other part. And so it's like, well, if you could generate sort of like, I mean, people try to like do this kind of thing with like evolution or whatever, where they generate a bunch of different sequences and see, okay, like what sticks around and what doesn't. But it's like, if ideally you could improve this thing, scissors, such a thing, like more precise, right? Like half the sequence, you just print out as like your guide. So
if I'm going to take the extremely optimistic case, then this research and its sort of impact, it's like we could have AI design special scissors, and that enables to do new kinds of gene therapies that like we might not have been able to do before.
Exactly.
Okay.
Yeah,
that's why.
Yeah,
I'm with it. Thanks.
nice. Good clarification. We care because it can help potentially create better therapeutics.
Great. A couple of YouTube questions I'm happy to go into this. Why not?
Yeah.
One actually I liked was Are there any benefits from applying these techniques, these prediction techniques, to the non-coding regions of the DNA? Right, we're focused mostly on predicting the coding regions, but is there any value in trying to predict non-coding regions too?
Oh yeah, absolutely. I think it's a space that, yeah, perhaps we could have focused even more on. And one reason for that is the long range interactions occur more often in these non-coding regions. So because they have this regulatory effect. And so yeah, I think that's actually the focus of the first paper on Hyena DNA. We actually focus mostly on the regulatory regions, modeling those types of sequences, but more on discriminative tasks. There's folks, this project called DNA diffusion that focuses on generating sequences, generating DNA for regulatory purposes. And I think that is indeed a very promising area. They did focus on like shorter sequences, so there's still definitely room on having long range interactions and being able to model those and generate from that kind of representation. Since there's a limit to some of these mini models, because some of these interactions could be millions of base pairs of nucleotides away. And so very important area to keep working on.
I guess related to that question on that point about like the context lengths, like I guess like how do you think of like I guess pre-training on longer context? Like how do you think that like helped? Because I think like there are some instances where, for example, like on the essentiality test, like the 8K to 66K, like the violin plots didn't seem that different. And also like with the CRISPR-Cas9 sequences, right? Like I don't think those were like within the realm of like 132K, like or you need like 128K like context length, right? Like it's existing models that did like I don't know 8K or whatever before good.
Yeah.
Those as well. Yeah.
Yeah. So yeah, I think long context is just one of those things that seems like it's like a sliding scale on how good one can be. Even though you have long context, it's really hard to tell like how good it is or how needed or how much it improves tasks. I think this is something you see even in natural language benchmarks. Being able to have good ways to measure how good a model is using its long context is a non-trivial thing. In our case, though, yeah, I think what we, I guess, mostly focused on was, can it maintain quality with long context in some of these zero-shot tasks? But then primarily for generating is what we were sort of driving toward. It's like, can we generate a whole genome, right? And so we can get to that scale in terms of the sequences generated. But we do still have that gap in terms of actually having high quality generations, right? And so it's, in that sense, maybe not necessarily what we're trying to optimize, not necessarily longer context, but just improving quality of the context we do have. And that's how my headspace is currently in.
I'm curious, like so for like the let's say like the Cas9, I guess like generations, like did you ever like I guess you had like 2 stages of training where you had the 8K model and then you had I forget 132K or 128K.
131, yeah.
131K, yeah. So like did you ever do, let's say, comparisons between the model generation quality with the 8K model versus the 131K, given they give both support, let's say the whole CRISPR-Cas9 like sort of sequence?
Yeah, that's a good question. We didn't. It's sort of like, I mean, we could. You're right. So we could just generate something 8K on the 131K. We didn't do that, I guess, because I suppose like there's like an efficiency thing, like efficiency in the sense that if a model trained on one sequence length, we probably our hypothesis is probably going to be better at generating that sequence length versus a model that's longer. It's probably not going to be as good as generating the shorter sequences. We didn't confirm that, but that's sort of like our intuition. Like, why use a longer model if you don't need to? Type thing. You most likely will hurt performance. But it's a fair question.
One more question I had for you. So I remember there was the plot about how the models trained on prokaryotes don't generalize that well onto humans. And I mean, I think this makes some sense that it seems like the underlying biology is fairly different in those creatures. Do you have thoughts about, whether it's worth trying to build foundation models that kind of span all these organisms, or whether you want to have more specialized, a foundation model trained on just only a lot of human DNA so that it's not, distracted by like the fact that fish work really differently to humans or something like that.
Yeah. Oh, that's like my favorite question. Because I think perhaps people have different opinions about this, right? Like actually probably a lot of folks that I've talked to seem to believe that a specialized model for each of these types or species would be better. But I personally, maybe driven by curiosity or just belief, I would absolutely love to see the large model capture mini.
Just make it bigger.
Just make it bigger. Because right, like looking at things like ChatGPT, right? You train things on math, train things on Chinese, you train so many different domains, even a lot of different things. So I just feel like this could be done in DNA as well. There's also some things going for it, right, that are in favor of this, which is that we do have some shared ancestry, right? So we do have some shared DNA with some of these other species, especially some of the simpler ones, simpler organisms. And so there's some arguments there as well for why it makes sense to combine it all. But there's plenty of reasons that people argue that it doesn't make sense. So open question still, depends on what you want to drive, what your beliefs are.
Yes, yeah, so intuitively then, they're like, how did you characterize the distribution shift between, let's say, prokaryotic versus eukaryotic DNA? Yeah, like, do we think about it as like the same as like the difference between like natural language modeling math versus Chinese, or yeah, like why was there, I guess, I mean, another way to think about it is like, why was there such a big drop in performance, relatively speaking, with like the when evaluated on like the eukaryotic, I guess.
I don't know how to make the analogy of how we think about the differences in the data distribution, but
I guess, but just like what's different?
yeah, the main difference that folks in that I talk to in biology, that they say that you need to carefully consider how you train these models, especially if you want to do eukaryotes or like humans, is these coding versus non-coding regions. So genes versus regulatory regions, because again, the coding regions have this basically deterministic mapping to what they map to in terms of proteins. The non-coding regions are way more multifactorial in terms of how they interact. There's basically non-linear interactions that compound that we don't know how they regulate the way they do. And they're cell specific also, so they behave one way in one type of cell, like say your brain versus your liver. And they will differ between species perhaps as well in this regulatory region. In the coding regions, they actually don't. Like the proteins map to the same proteins across cells and across species, which is pretty wild. So if you're going to go from a species that is mostly coding regions, like these prokaryotes, to humans, which is mostly non-coding regions, then you need to start thinking about how your models that could be confounded, basically.
Right, I guess as you say, this sort of makes sense to me that if you were to try to just train naively on human DNA, you'd have this problem that almost all of the loss comes from this like huge spikes up, just tons and tons of total at least what we think is noise in the non-coding regions, and then maybe that distracts the model. It doesn't do as good of a job of learning stuff that you care about a little bit more, which is the coding regions. At least on a per token basis, you care a little bit more about the coding regions. So you end up hurting your performance a lot. That totally makes sense. That would be a problem.
Yeah, exactly. And so I think that going into this EVO project, people thought that would be the case even for prokaryotic species. They're like, you're going to train on both coding and non-coding, and expect the model to be good at the coding, as good as protein models, which is like got this advantage of just seeing proteins. Most people were like, that's not going to work. And in hindsight, I guess some people were like, oh, obviously it's going to work. Like these prokaryotic species are mostly coding anyways. I would argue that if it was so obvious, no one had tried it and tried to show that it worked. So I think that's actually, I love to hear those kind of comments. Like if it's obvious in hindsight, I love it. I love that kind of thought.
So maybe it'll be obvious in hindsight that, of course, trading just the foundational human DNA would have worked. The coding regions have some structure, the non-coding regions also structure. Like we just didn't understand it, but the model figures it out. Maybe that's what it'll be.
Fingers crossed. Yeah, we'll see.
We'll see. Any other questions, Michael?
You can wrap it up.
All right. Well, thanks so much, Eric. This was a blast.
Woo, that was fun.
And we'll be back next Wednesday with our next speaker. So thanks for tuning in.
Thanks, guys.
Bye, YouTube. All right.